This method can provide important information related to the pupation ecology of tea loop caterpillars, Ectropis grisescens, such as their pupation behaviors and pupa survivorship in response to different environmental factors. The main advantage of this technique is that it uses simple methods to determine the pupation preference and emergence sexes of Ectropis grisescens.
To begin, cut fresh shoots from tea plants. Then fill a 250 milliliter triangular flask with tap water and place the shoots into the flask. Transfer 3 to 4 flasks to a plastic basin. Next, release 1,000 to 2,000 larvae from a laboratory colony of E. grisescens onto the leaves of the tea shoots. Maintain the larvae in controlled laboratory conditions. Obtain the mature last instar larvae that fall from the leaves of the shoots and actively wander along the bottom of the basin. Sterilize soil and sand in an 80°C oven dryer for 3 days. Then dry both the soil and sand at 50°C for several weeks. The substrate samples are sufficiently dried when the weight does not change over time. Use a wooden pestle and mortar to grind the dried soil. Then sift the sand and the ground soil through a 3 mm sieve. Add the required amount of distilled water to the sealed bags containing the substrates and mix thoroughly. Next, use PVC sheets to divide polypropylene containers into 6 equal chambers. Fix the PVC sheets and seal the cracks with hot glue.